For this figure, this goes through the different types of means to preventing a fertilization event and at what stage it's able to impede that process. So we've talked about permanent and temporary methods. And one thing that we didn't talk about was the idea of abstinence, which is a behavioral method. And so the abstinence, notice where we find it within the process, it doesn't impact the ability to produce sperm or oocytes or to have sperm go through the duct system, but it does prevent the likelihood of sperm and the oocyte ever coming into contact with one another. So this would be a behavioral approach that is within the figure, but wasn't discussed in the earlier image. And coitus interruptus, which is essentially trying to avoid, even though the sperm is transporting its way down the duct system, avoid actual deposit into the vagina. Very high failure rate. This should not be the means in which individuals try to avoid pregnancy because it's just really inconsistent. You can't really do this as effectively as the other methods that are found within this figure. So what you want to do when looking at this figure is think about how the male and female reproductive tract are being utilized within this process, which types of methods are applicable to one over the other. So meaning, a vasectomy would be specific to the male reproductive system, tubal ligation specific to the female reproductive system, and then just kind of thinking about what impact it has, how far down the pathway are we able to impede the process of what we see all the way at the end, which would be implantation of a blastocyst in the endometrium. So I'm not going to go through this specifically because we've already gone through these methods, but and we've also already gone through the process of the events that ultimately lead to the implantation, but just think about how they connect to one another and the timing of them. And for the most part, and this isn't really applicable to everything, but, as you go higher upstream in terms of, preventing these events, that increases the likelihood that we can avoid that implantation event from happening, right? So if we have the vasectomy and you are able to produce sperm because of that vasectomy, the ability for that sperm to transport its way through the duct system is impeded, which means if it can't do that, we can't achieve any of those downstream events. Now, if there is the implantation event that occurs, then abortion can be a termination of pregnancy once that has taken place. Now, spontaneous abortions are also referred to as miscarriages, and they can occur in a pretty high fraction of pregnancies. And when it comes to these abortions, sometimes these would be a result of the organism realizing that there is going to be aneuploidy or something has not formed in such a way that it can sustain implantation successfully for an extended period of time. There are tons of reasons why they can happen, and a lot of them take place really early on within the first trimester. But there can also be drugs that can achieve this same idea, which is to terminate a pregnancy. And it's a non-surgical procedure, the one that's shown here. And the idea behind the drug is that it creates essentially an inhospitable environment for blastocyst implantation. And so as a result, the kind of uterine lining is going to be shed. And in doing so, there's not going to be anything for the blastocyst to implant itself into and receive nutrients from. Now, in vitro fertilization is an example of a reproductive technology which has the capability of helping to assist individuals that may deal with infertility. And that can be something that's seen on both the male and female side as well. They can be equally affected. Now, in vitro fertilization, or IVF, would be external fertilization followed by an embryo transfer. So the eggs and sperm are going to be within the culture dish, and then the embryo gets returned to the uterus at around the 8-cell stage. And then from there, the uterus will take over. So, not comparable to like the Dolly the Lamb setting as far as like the setup goes, right? Because we know that that's a somatic cell nuclear transfer, but similar in the sense that we are manipulating something outside of the organism and then adding it back into the individual that's capable of carrying that cell into something that will differentiate and change into a new organism. Now, a downside to this process, at least right now, is that multiple births are pretty common when in vitro fertilization is used, because it's really common for them to transfer in more than one embryo, because the goal is to ensure that at least one implants and develops normally. And so oftentimes more than one will implant and develop normally. So, working on how to ensure that this isn't the case is something that would be beneficial from a reproductive technology standpoint. So, with all of these different patterns of inheritance and ways of passing on information through autosomes, through sex chromosomes, we can gain a lot of insight about an organism's development through analysis that can occur during fetal development. So Fetal cells can be analyzed through different mechanisms. Here you have two examples of amniocentesis and chorionic villus sampling. And when it comes to CVS, this can be done earlier in the process. So notice that it says 8th to 10th week of pregnancy, whereas for amniocentesis, it's a little bit later, so 14th to 15th week of pregnancy. But in both instances, the goal is the same, which is to acquire fetal cells, because within those fetal cells, we have information related to genes. And so we can sample those cells and then ultimately generate a karyotype. And again, a karyotype is a way of organizing chromosomes so that we can look for any abnormalities, any extra chromosomes or chromosomes that are missing, chromosomes that may be partially deleted or translocated, moved to another position. So just in looking at the method between these two, for amniocentesis, you have a needle, it gets inserted into the amniotic cavity, and a sample of the amniotic fluid is derived from the fetus. And you'll take that sample, you'll place it into a tube. We can centrifuge it so that we can isolate the components that we want, right? So the least dense will settle in the top, the more dense will settle down in the bottom. And what we want is those fetal cells. For the CVS, it's the same thing in terms of acquiring fetal cells, but the location of acquisition is different. And so in CVS, the cells are removed by suction with a tube that's inserted through the cervix. And again, this one can be done a little bit earlier, and the data can actually be obtained a little bit faster. But this methodology can increase the risk of miscarriage. Both of them come with their own risks, but if we're comparing them, this is a little bit higher. Now, in both cases, we're taking the fetal cells, we are culturing them, so we're placing them into a dish, and we focused on setting up karyotypes in order to understand what's going on with the chromosomes, but they can also be subjected to biochemical tests and other genetic tests, and this can really be used to assess the genetic state of the fetus really early on. And so this information can be really beneficial in terms of advising parents on medical treatments or options if a genetic disorder is determined to exist. And this is the benefit to genetic counseling, and that would be a process that can be used by expecting parents in order to identify any possible risks of producing children with genetic defects. So in looking at this particular slide, just think about both of these methods downstream can serve the same role, which is to kind of generate genetic information that can be used to determine the likelihood or risk of an organism developing with some type of issues or abnormalities. So compare and contrast them. Spend a lot of time on the compare, because that allows you to go back to the earlier discussion of karyotypes and how that is relevant to gleaning information about just the genome and the integrity of that individual's genome.